Hello and welcome. Thank you so much for joining us. And today, as my guest, I have Zach Hancock. Welcome, Zach.
Hello, thanks for having me.
Thank you to everyone who is watching in the chat and those who are watching in the future. This should be a really great show, and I'll tell you how this show came about because it's kind of a funny story. I was on Twitter and I was talking to someone who was arguing in support of answers in Genesis. And they said, well, evolution, it has limits to it. Speciation can occur, but obviously that there's a mechanism that stops from going beyond a certain point. And I said, well, what is that mechanism? How have you identified this mechanism? And they posted a paper. Now, the paper doesn't actually discuss any kind of mechanism or support his claim that there was a mechanism, but the paper claims that evolution beyond just change of species is impossible statistically. And so the first thing I did in examining this paper was to look at who wrote it. And it was written by an engineer, not an evolutionary biologist, which I said, okay, that's a little suspect that an engineer is writing about evolutionary biology. But on the other hand, the paper primarily dealt with probabilities, and he was analyzing the probabilities of whether or not evolution could occur. And I did not have the entire paper. I only had the abstract and what you can get on the free copy. So the next thing I wanted to do was look at any peer reviews of this paper. What were the professionals saying about this paper? Because that would be a lot easier for me to do. And I couldn't find anything. And I wasn't sure if the reason I wasn't finding anything was because nothing existed or because I wasn't doing the search properly. So then I turned to a friend, Jackson Wheat, and I asked Jackson Wheat, am I doing this search incorrectly? Are there any peer reviews on this paper? And Jackson Wheat said, oh, you should talk to Zach Hancock about this paper. And so that's what I did. I contacted Zach Hancock. I asked him about this paper. We decided to go ahead and do a live stream and address this paper together. I still do not know the answer to the questions that I had about this paper and what is wrong with this paper and why it does not completely disprove evolution. And that's how this show came to be. Welcome, Zach, and we are looking forward to what you have to say.
Thanks for having me. I'm excited to talk about this.
Okay, let's start. I'm going to go ahead and pull up the paper. And there's a couple of portions of it that I have highlighted. And let's see. Have I got the right? Did I get? Nope, that's not it. Sorry. Let's try again. Anyway, I'm going to pull up the paper and we'll talk about some of the things that I've got highlighted on it. There we go. Okay, is that visible?
I can see it.
Okay, very good. All right. This was the first thing that I highlighted on it, was he was saying something essential is missing in the theory of biological evolution, neo-Darwinism. He says any overall mechanistic explanation of the origin and evolution of life ultimately must satisfy two challenges: the transition from non-life to life, and the blossoming of life forms that is so extreme as to appear outrageous. Now, my first impression from that was, wait a minute, evolution doesn't deal with the transition from life to non-life. It only deals with the transition from life to life. Am I correct in that assessment?
That is correct. So every summer I go to an evolution meeting, and it's a meeting where thousands of evolutionary biologists from all over the world, we come, we meet at a conference, we all give talks, we present our work. It's a lot of fun, but at none of those meetings is the origin of life ever discussed. It's not a topic that any of us work on, because it's not a topic of evolutionary biology. It's a topic of organic chemistry. It's a topic of biochemistry. The people that work on it are chemists. Most of them aren't even biologists. So it's since it kind of straddles the line between biology and chemistry, evolutionary biologists are very far removed from it. We're interested in how the diversity of life came about and the mechanisms that generate that, and the origination of that diversity. We're not interested in general in when life began. As far as many of us don't even really care. Like, God could have poofed life into existence and then everything evolved from there. Doesn't make a difference to us. We only care once it's there to start going.
Or Lisa the rainbow giraffe.
Yeah.
Are you familiar with the skeptic?
Like, is that like a name?
He's a YouTuber. He has a channel called The Skeptic.
Oh, like a
and he's the floating British circle. And he invented Lisa the rainbow giraffe, who pooped the world into existence. And one of the things he always says is Lisa the rainbow giraffe, and you always say "leaf be upon her" when you use her name.
Okay, I'll keep that in mind.
Uh,
There was something interesting that you brought up, sort of introducing this, that may be worth chatting about before we dig too far into the paper, is why there was no responses to this paper.
Okay.
And I think this is a really important point. So this paper was published in Progress in Biophysics and Molecular Biology. Is it the full name of the journal? Progress in Biophysics and something.
Let's see.
But this journal, and I nailed it. All right. Yeah, Progress in Biophysics and Molecular Biology. And this particular journal is run by a physiologist named Dennis Noble.
Okay.
Dennis Noble is, for all intents and purposes, a little outside of the mainstream. He's a bit of a fringe guy that has lots of kind of wild takes. And this particular journal is kind of infamous for publishing wild takes. You might be familiar with a paper from a few years ago in which it argued that cephalopods, that's like octopus, squid, etc., are from outer space. So if you
yes, I did hear about that one.
So that paper was in this journal. And so that just kind of gives you a bit of a feel about the kinds of papers this journal will publish. Dennis Noble is a ardent critique of what he calls neo-Darwinism. We can talk about what that means, but he's an ardent critique of that. And so basically any paper, any authors that will come out and kind of critique that idea, you can find their stuff here. So that's one of and because of that, it's not a highly respected journal among evolutionary biologists. We don't read it. We're not going to waste our time responding to it because it doesn't publish respectable work. And so it's difficult for like a lot of people that may not be in academia to know that there are a ton of journals out there that will publish basically anything. There are many what we call like pay to play journals, where so long as you can pay the publishing fee, they will publish a paper. And it's kind of a con, right? Like, and a lot of really bad papers can get out there because of this, including like creationist papers, right? So you have to be careful whenever somebody sends you a paper like this, and it's like from some journal that maybe you've never heard of. Take some time and Google that journal. Look up what's called the impact factor. Usually if it's not above 4 or 5, it's a little suspect. And then also see if you can find a Wikipedia page. Who are the editors in chief? That sort of thing. And that will give you a little bit of an idea about whether or not this is a legitimate journal.
Okay. What's the impact factor?
The impact factor is how often the papers that it published are cited by other people. And so so that's like the impact that the paper has, right? So like if a paper has been cited by thousands of researchers, and it really made a big impact, right? But if your impact factor is really, really low, then it's like a ratio of citations to publication sort of thing. Then that means that these papers aren't making an impact. People aren't reading them. And that tends to reflect quality. Not always, but it tends to reflect quality.
Okay. All right, the next thing that I highlighted from this paper was, let's see. Okay. It's beginning mid-sentence here, so I'm thinking I'm wondering if I grabbed, okay. He talks about probability of evolution. Probability, like any scientific analysis, has limitations. Because evolution is generally accepted as scientifically established, probability assessment has largely been overlooked. Evolutionary probability generally is said to be supported by the statement that billions of years make evolution possible. However, this overlooks the fact that time is a linear factor, and evolutionary probability inevitably involves exponentials that are overpowering in effect. To say a probability calculation is valueless because evolution has already occurred is a fact is mathematically flawed. Probability calculations pertain to future events or the number of attempts for the event to occur. To say that it occurred without knowing how many attempts have occurred has no meaning. And the reason I wanted to discuss that is I have heard that argument. Oops, I forgot to put that on screen, didn't I? Sorry about that. While I was reading, I thought I had that on screen while I was reading. I apologize for that. I've heard people make this argument that, well, something has already happened, so the probability of it happening is one because it did happen. And so I do get what the paper is saying from that we're not looking so much at what is the probability that it could happen, because obviously something did happen, but does natural evolution explain, is the probability of evolution being correct a proper explanation for what we see in evolution? So I get what he's getting at. But the big question that I had as a result of reading that was, well, do we know the factors that he's talking about? Specifically, he says, that such a probability, calculating such a probability is meaningless if we don't know how many attempts have occurred. And I'm thinking, well, we can't possibly know how many attempts have occurred. We can't possibly know how long the universe could possibly have attempted to and failed at any number of biological, I don't know, developments, I guess is the best word. What
Yeah,
are your thoughts on that?
yeah. So this whole passage is a little nonsensical, and I'll try to break down as to why. So for one, it's a creationist talking point.
Right.
We don't calculate probabilities in this way for evolution, and we don't rely on time. Like nobody says, well, we don't need to calculate it because of time. That's not how evolutionary research is done.
Okay.
And there's a key reason for this, is that the particular probability that they are trying to do, and he uses the example of how can the Krebs cycle emerge. That's what he uses later on. And he says, okay, if there are X number of enzymes within the Krebs cycle, each of which are composed of some number of amino acids, then we know all of those amino acids have to be there. So what's the probability? And there's 20 possible amino acids per site. And so what's the probability of all of that emerging in effectively one dice roll? Right? Like, that's what he's asking, is all of that has to emerge. And he's making several really important assumptions here. One, he's assuming that each of these are independent of each other. This kind of probability calculation assumes that every single roll is independent, so the probabilities are independent. And it assumes something really important, and that is that evolution is trying to get here, right?
Yes.
And so if I look at a protein that exists today, and I say, okay, what's the probability that this protein occurred? And I want to calculate that probability as the length of the protein and the number of possibilities for each site, then any protein, the probability is going to be infinitesimally small, right? But that's true for any number, any kind of string of numbers in which you draw randomly, and the longer the string is and the more possibilities per site, the more inevitable or the less probable it's going to be, right? And that's for any number. But that only matters if you assume that this was the only protein that could emerge, and it's the only one that evolution was trying to get to in the first place. We know that this is nonsense. And the reason we know this is nonsense is because the biological world is full of diversity. And it's full of diversity because there's actually a ton of ways to build organisms. There's a ton of proteins to land upon, right? There's not just one. We all are built of proteins that are composed in many different ways. So to presume that evolution had to find just one is preposterous, right? So I call this hindsight evolutionary thinking, where it's you see something in the present and you say, okay, how likely is it to have occurred from the past? When really there's an entire protein space. Right? If we imagine all the possible combinations and you just drop the beginning of the evolutionary process, somewhere in this protein space, there are many different avenues it's going to walk through, and this represents each lineage that is testing out different places in this space, and some are going to land on useful combinations and some will not. The ones that land on useful combinations proliferate, right? And those that don't go extinct. And this process is repeated many times. And as you explore this protein space, you find there are tons of places in this space that will generate function, right? It's not that there's just one possible direction you have to get to. So the question itself is malformed, and it's kind of lodged as a sort of insult. They're like, well, they just assume, and you hear this all the time from Ken Ham and people like this, they just assume millions of years, and that way evolution must happen if there's millions of years, whatever. But like, no, we don't think like that. We don't sit around and be like, oh, well, there's enough time. No, we want to know how it happened. We want to know the mechanism. But we don't calculate probability in this way because we don't apply goals or teleology to evolution, right? This is an incorrect way of thinking about evolution. And this entire paper is kind of, or at least the probability portion is predicated on this malformed idea that evolution has a goal.
Which is really interesting because right before we came on, one of the things that I said to Zach is, if I don't ask a question correctly, please feel free to reform the question for me. And you're telling me that's exactly the problem that is with this paper, is they haven't formed the question correctly.
Yeah, that they are attacking what they think is a problem that's not a problem. It's a problem of their own creation, right? Lawrence Moran has this great book called What's in Your Genome? And there's this quote in it where he's talking about paradigm shifts, where like molecular biology is causing a big paradigm shift. But oftentimes what you find are what he calls paradigm shafts, where instead of actually representing the paradigm as it is, you misrepresent the paradigm. And then you say, oh, I've overturned this misrepresentation of the paradigm, right? It's a paradigm shaft. And that's what they're doing here, is they're misrepresenting evolutionary research. They're misrepresenting the way we think about evolution. And then they're saying, look how wrong they are with this misrepresentation.
Mm-hmm. He goes, and I'm going to summarize the paper here because it takes him a couple of pages to describe this, through an example of how if you were on a deserted island and you were going to build the Empire State Building and you were given one piece of the building each day, and if the piece that you were given doesn't fit where you are on the building on this particular day, that piece is taken away at the end of the day. You would never statistically end up building the building because you're not going to get the pieces in the right order. And from what you're telling me is that the whole illustration is incorrect, in that there is no goal of building the Empire State Building.
Exactly.
That might be what you ended up with, but as you're given the building blocks, kind of like with Legos, you could end up with anything at the end.
Yeah, and then you can imagine whatever you end up with at the end, and then all the possibilities, all the directions you could have gone, and then say, well, what's the statistical probability I ended up with what I did? Well, it's going to be really, really small because of all the possible ways you could have gone. But you're going to go one way. Like there's some direction is going to be taken. And so that's the problem with this hindsight thinking, is that once you see the thing completed and then ask, well, given how many possible directions you went, what's the likelihood you went this way? But you're going to go one way.
We have a question here from Nitty. Different nucleotides combining can produce the same proteins or foods, right?
Yeah. Okay. Yeah, they can produce, and they're not like exactly identical proteins, but they have the same function. I think that's what you mean, is that despite many different ways of combining those nucleotides, you can produce a protein that does the same thing. And there's also ways to produce basically the identical protein in terms of the amino acid sequence, but have different nucleotides underlying them because of the redundancy of the genetic code. So the genetic code is in triplets, right? So it's like ATC, GGA, right? There's these triplets that code for a particular amino acid, and there's some redundancy there. So you can get the exact same amino acid from different triplets.
And Nitty says, I meant functions. Yeah. Dang, autocorrect.
Yeah, I assume that's what you meant. Yeah, you can get the exact same functions.
I did not understand the question at all. That's how I was going, what? Because I know that there is protein in food, and so I don't think that's what he means, but I wasn't sure. Okay, great. Glad we got that clarified. Yeah. Okay, I'm going to pull up another portion of the paper. Let's see. Oh, here we go. It's just a little further. Most relevant to our use of probabilities is a contribution by Perry Marshall, published in 2021 in Progress in Biophysics and Molecular Biology.
This
We will
journal.
quote from this. Yeah, that I was going to ask you if that was a reputable paper, person, etc., but I guess you've actually already answered that question, haven't you?
Yeah. Yeah,
Because it's in this
it's
journal.
in this journal that he's referencing. And actually a lot of the references throughout this that are critical of evolutionary theory are in this journal. Like he references Dennis Noble many times throughout. Dennis Noble is the editor-in-chief of this journal.
Ah, okay.
Yeah. So there's a little bit of bootlicking to promote this particular work, right?
I was going to mention a different body part, but same idea.
Yeah.
Okay.
There's also like and this is a kind of a broader thing. They mention survival of the fittest. A lot of their critique
Right.
is survival of the fittest.
Mhm.
And survival of the fittest is not a phrase we use in evolutionary biology. Um,
Okay.
and there's a really important reason as to why survival of the fittest was proposed by Herbert Spencer, and Darwin later adopted it at the behest of Wallace, to try to make clearer what natural selection was supposed to represent. But
Mhm.
survival of the fittest is what we call a tautology, right? It's like you survive because you're the most fit. You're the most fit, therefore you survive, right? It's this kind of circular tautology. But that's not the way we actually view natural selection. Natural selection is a probabilistic. And this is why his critique that we don't use probability is really, really silly. We use lots of probability in statistics. In fact, most of modern statistics was developed by evolutionary biologists in the early 1900s.
Really?
Yeah. So the school of the biometricians, if you like just Google that afterwards, anyone interested, you'll find that people like Carl Pearson, people like Francis Galton, R.A. Fisher, Sewell Wright, they develop all of what we know of modern statistics was developed by these guys. So linear regressions, chi-square tests, ANOVAs, ANCOVAs, et cetera, was all by them. Yeah. So survival of the fittest. The problem with survival of the fittest is that there is a probability that any allele in a population goes to fixation by positive selection, right? And that probability is actually dictated by many factors of that population, including things like breeding structure, including things like population size. So the smaller your population, the more chance has a role in what allele gets fixed, right? So just because an allele provides a fitness advantage doesn't guarantee that that allele is going to fix. Survival of the fittest would state that yes, if it provides an advantage, it will always fix, and that's a tautology. Since we actually approach this probabilistically, it's natural selection is not a tautology because it's not guaranteed that the most fit allele actually is going to fix. So I think it's important for us to understand this distinction, and there's a lot more complex math behind natural selection that I don't want to get us into the weeds of, but that point being is that survival of the fittest is not a phrase we use. And so it's very telling that their critique throughout this paper is survival of the fittest instead of an actual representation of natural selection as we understand it today.
The biggest tell for me in the paper was talking about macroevolution and microevolution.
Mhm.
And he began the paper by saying, we understand that microevolution has been observed in the laboratory, but macroevolution is what we are disputing here. And I know that an evolutionary biologist would never use those terms.
Yeah. So to us, microevolution is effectively population genetics. It's effectively population quantitative genetics. We're looking at changes within single populations. Macroevolution to us is a broader field of study that is looking at patterns, right? So if you're interested in like building the tree of life, if you're interested in like community compositions, if you're interested in species interaction, like these sort of broader patterns, that's macroevolutionary research. But the mechanisms are the same. So there's this assumption I think that he's making, or that they're making in this paper, is that there's something fundamentally different between the processes of populations and then the processes that explain the origin of species, genera, families, etc. But those processes are exactly the same, and they're the same processes that we've known about since the early 1900s, right? And so it's curious that they're asking for this missing mechanism when there is none. We've already provided that mechanism, over 100 years ago.
Okay. Now, the mechanisms of evolutionary biology, if I understand correctly, are not only natural selection, but also genetic drift
Mhm.
and population.
So it's natural selection, genetic drift, gene flow, and mutation.
Okay.
You can also include recombination is sort of a very important mechanism that helps to shuffle genotypes that generate new phenotypes for selection and drift to act upon. But these mechanisms, basically, you have two of them that remove diversity. That's natural selection and genetic drift. They're always acting to kind of purge genetic diversity. And then you have the two other forces that are adding it in. That's gene flow and mutation. And so these two forces, or these four forces, are all interacting in complex and really fascinating ways, particularly selection and drift, because they're often opposing forces, like trying to pull populations in different directions. And so, yeah, so those are the primary mechanisms of evolution.
Okay. Okay. One thing that I hear frequently from creationists that I kind of know how to answer, but not very well, is they continually make the claim that evolution is incapable of creating new information.
Mm.
That yes, there's genetic mutation, but it's always just a repeat or a rearrange of the information that is already there. It's never new information.
Mhm.
How do you handle that question?
Yeah, so there's several different ways you can approach this. I've seen people try to pin them down on what information is.
Mhm.
That's not really a route I go. I just like try to assume that by information they mean something like like a phenotype, like a genotype that codes for a phenotype. I assume that's what they mean. I try to give them the benefit of the doubt and not try to squash them on the definition of information. So if we assume that by information we mean generate something new, something novel, either a novel protein or a new biochemical pathway, something like that, right? We take that as being information. Then it's very easy to refute this because we have so many examples of this exact thing happening. And we even know many different ways in which it can happen. So at the very basic level, we have what's called de novo gene synthesis. And this is where you have a non-coding region of DNA. That's a region that doesn't make anything, that then through subsequent mutations acquires what we call an open reading frame. That's where you have a start codon and then enough bases down and a stop codon. And this basically is what you need to tell, the ribosome start coding at this end and stop coding at this, and then it will generate an amino acid string, right? And this can happen completely randomly in non-coding sequence. And it's actually really easy to pick these up. So what we can do is we can take two closely related species. This is famously recently done in yeast. Yeast is perfect for this because we have baker's yeast, that's sort of like the domesticated yeast. We really understand their genome super well. And then there are wild yeast, right? There are like other species of yeast as well. And so we can go out into nature, we can take these wild yeast and say, okay, what genes do they have that we don't see in baker's yeast? And since we know that they are very closely related yeast, that any genes that the wild ones have must be novel. They must be some new gene.
Mm.
And so what we have found is that there are populations of wild yeast in which these brand new de novo genes are polymorphic. That means some individuals have them and some individuals don't. And this tells us that they must have emerged and they're spreading through the population but haven't become fixed yet, right? So we have like sort of an empirical example of novel genes emerging. And they have really cool characteristics. So very often de novo genes are very short. They're often pretty simple because they haven't had time to like become elaborate. They usually have a little bit lower transcriptional efficiency, which means that they're not like transcribed like normal genes quite as efficiently yet because these things take time to build up. But we see things like selection acting on them. We see them behaving in functional ways with other protein interactions. So we know that they're doing things for the organism, and we know they're spreading through the population. So that's sort of a de novo from non-coding gene. This is a bit of a rarer sort of way of getting new information, but it's literally new information from things that didn't have information previously. A bit of the more standard way is by gene duplication. So you basically have some existing information, you duplicate that information either in tandem or by recombination to some other place. You have now two copies of it. And since you now have another copy, it's freed to explore mutational space, right? Because selection is keeping that single copy constrained, right? Because if it mutates and breaks, then you can't do anything with it anymore. But if you have two copies of it, now one of those copies can become is more mutationally permissible, right? And so there's a couple of different fates that this new copy can take. The new copy can either undergo what we call subfunctionalization. And this is a really interesting way of generating complexity in genomes. So if it undergoes subfunctionalization, what that means is since you have two copies, some mutations can occur in one and in the original one, such that they're both degraded in some way that they now rely on each other. And this actually creates what's called epistasis, so interactions between genes, and they're both necessary to be there. And we believe that a lot of the complexity in eukaryotic genomes were actually generated by this process, that you have lots of duplications, they each kind of mutate in their own way, and now they're all like linked in this complex web of interactions because of this subfunctionalization activity. So that's one way. But another way that they can actually that they can gain new information is what we call neo-functionalization. So the original copy stays normal, keeps its original function, and the duplicate now acquires a new function. Right, so it gets a mutation that now interacts with proteins in novel ways. It has different binding affinities, etc. We have lots of great examples of these as well. The best studied are in Drosophila, because that's a fruit fly that we've studied in labs for over 100 years at this point. And so we very well know what their genomes are like. So we can take sister species and we can say, okay, these are duplicates in each of these species that are unique to these species. Do they seem to have novel functions? Do they seem to have novel binding affinities relative to the parental copies? And we have hundreds of examples of this. So we know neofunctionalization happens, we know subfunctionalization happens, and that's a really simple way of increasing the information content in the cell.
How do you learn that? And what I mean by that question is, how do you learn what function a particular gene has?
So this is there's these kind of molecular assays in which you can take some protein structure because you know what it codes for, and you can synthesize this protein. Right. So if you know the amino acid, you can synthesize it in a lab, and then you can subject it to different substrates. So you can say, okay, does it bind to this substrate? If so, what's the affinity? How well does it bind to this substrate? You can also find out function by knocking it out. So if you have this neo-functionalized region, you can just like snip it out of the genome and then say, okay, how does development happen? Does anything change? Do we see a pathway fall apart, right? And that will tell you, like directly, okay, well, this was serving an important enzymatic, process within this pathway. And so we know that by shutting it off, it must have been, like we've now broken it, right? So by either breaking it or by actually subjecting it to a molecular assay, we can determine what these proteins are doing.
That is so cool.
It's pretty neat, yeah. A third way of generating new information, and this will be the last one I'll say.
Oh, that's all right. Go ahead.
Is by what we call retrogenes. And retrogenes are really cool. So if you take a protein, an mRNA comes and they transcribe it, and so you get this mRNA transcript. Well, that mRNA transcript is currently what we call pre-processed, which means it still has introns. So in eukaryotes, their protein coding regions have exons and introns, and the introns have to be cut out, and then all the exons are snipped together, right? So actually your genes are bloated with things that have to get cut out. And so that happens after the initial mRNA transcript is formed. Now, once you get a post-processed mRNA, where you've cut out all those introns, sometimes those can accidentally get reinserted back into the genome by reverse transcription. And so you have the original copy in one place, and then it gets, you make the transcript, it's supposed to go out and be turned into a protein, but it gets picked up by what we call a reverse transcriptase. It's just an enzyme that will force it back into the genome. And it just like adds sort of like a duplicate copy. Most of these become what we call pseudogenes. So they degrade, they don't do anything. A lot of the junk in your genome are these sorts of things, they're these retro elements, right? But every once in a while, they will actually acquire a secondary function. And we can tell that they've acquired a function because they're transcribed at really high levels and because they're sequence constrained. And that means that when a mutation happens there, it's deleterious, and so selection gets rid of it, right? So it's kind of maintaining that sequence identity. So those are sort of the three big ways in which we understand how new information gets added to the genome. And again, I can't stress this enough. We have hundreds of studies showing these processes in labs and in nature. This is an incredibly well documented process that when creationists say mutations don't add new information, I'm just baffled. It's just like, it's like looking at the sky and saying it's green. I don't know how they can do it.
Maya Atkinson says, this sounds complicated. And I totally get you, Maya. As Zach was giving this answer, I was thinking I need to go through this stream after we're finished and cut this portion out and make that into its own separate published segment. Um, how we know that there is new information in evolutionary biology.
Yeah, it is complicated because there's a lot of like genetics and molecular biology underlying this, and it's sort of hard to explain this without using the jargon. Like I try to not use the jargon, but yeah, it just seeps through. But yeah, basically the takeaway is you can either get new genes from regions of the genome that didn't make genes originally. You can get them by doubling, just duplicating your gene and then having one of them turn into something new, or you can get them from a transcript of one gene gets reinserted someplace else and then gets co-opted. Right? That's the basic take home.
Very good. Very good. We have a question that I've actually been holding for some time. So this goes back a bit to what we were talking about earlier. I've heard of sexual selection, individual selection, and kinship selection, and some have talked of group selection, two of the species or group. Do you think selecting a herd is a herd thing?
Okay, so this is one we can really easily get into the weeds on, and I'm going to try very hard not to. But basically, let's try to break this down really simply. So selection typically acts on individuals, right? and it's because it's acting on a phenotype, and a phenotype is coded by a genotype, so it's acting on the individual phenotype. Now, when you have a population that's subdivided into groups that are like migrating between, they're not freely interbreeding with each other, then what you can have is you can have a group level trait emerge, right? So it's that everyone in this group has this trait, everyone in this group has this trait, right? And so what that means is that you can have selection happening not just on individuals within the group, but you can have selection acting on traits at higher levels between groups, right? And now this is complicated because it makes several important mathematical assumptions that are not always held in nature. We can make models of this pretty easily, but how often it occurs in nature is a little suspect. And the reason for this is that the efficiency of natural selection depends on the strength of genetic drift. And genetic drift is the random component, right? So more individuals in the population, the more efficient selection is. The smaller the population, the less efficient selection is. Now, at the individual level, in any kind of group set, any kind of population, even if it's subdivided, there are always more individuals than there are groups, right? Because like the group number must be less than the total number of individuals in the population. And so what that means is that at higher and higher levels of biological organization, such as like between groups or between herds, there are fewer herds than there are individuals, and so selection is less efficient. So to make the math work, you need all of the groups of the herd to breed true. They need to like only produce their group phenotype, and you need many groups. So that's really the only way that selection at the group level has been shown to be mathematically feasible. I know that's kind of a dense answer, but it's a really dense question, to be honest. It's a really complex question that has been debated for a very long time in evolutionary biology. So I hope that answered it in a way that was as clear as possible.
I hope so too. Let's see. Okay. All right. I'm going to go back to the paper one more time, but this is the last of what I had highlighted in the paper as being of question.
Mhm.
As the majority of the literature in biological sciences takes the capacity of life to evolve for granted. Despite detailed investigation of many evolutionary mechanisms, our understanding of evolution is very superficial and incomplete. Furthermore, the mystery of what drives adaptations is deepened by the phenomena of evolvability, the hard problem of life squared. Modern synthesis proposed that random copying errors combined with natural selection and population genetics would provide a gradual increase in speciation. There are several problems with this view. One is random mutations are noise, and noise destroys information. Evolution is not steady and gradual, it instead consists of long periods of stasis followed by rapid stages of punctuated equilibrium, contrary to Darwin's predictions. Because future events are undecidable, let's see, because future events are undecidable propositions, in principle, no algorithm can exist that will guarantee any future goal can be reached. Goals are central to evolution. Well, we already dealt with that part.
Yeah. There's a lot to that. There's a lot in that.
Yeah.
I don't even know where to begin. There's a lot of weird conflation and things that don't quite make sense, such as the conflation of Darwin's theory with the modern synthesis, which are different things. And the idea that evolution, like you said, evolution has goals, or the idea that punctuated equilibrium is somehow counter to our understanding of evolution, as if it's something we didn't expect to see. There's a lot there. Did you have a particular thing you wanted me to focus on in this?
No, you go wherever this one takes you.
Okay. I think maybe a useful place to start is with punctuated equilibrium, what it is and what they meant by it.
Okay.
So the idea of punctuated equilibrium was proposed by Steve Gould in the early 1970s. Steve Gould and Niles Eldredge. And they basically looked at the fossil record and said that, well, most of the time in the fossil record, we see stasis, that is very little morphological change, followed in the next layer by very rapid change, very rapid turnover of traits, of species traits. And they called this punctuated equilibrium. And they basically lobbed it at the modern synthesis as evidence that gradual evolution didn't occur, that evolution happened in sort of fits and starts with long periods of stasis and then rapid change. This was a very controversial view at the time. It's a very controversial view today. And the reason, and I've actually published on punctuated equilibrium. I've critiqued it in the scientific literature before. But the problem with punctuated equilibrium is it has several assumptions that don't hold super well in nature. One is that when you're looking in the paleontological record, when you want to designate a new species, necessitates morphological change. You can't tell if it's a new species unless there's some morphological difference. And so as soon as you see morphological difference, you say it's a new species. And so by necessity, change is punctuated in the fossil record, right? Because like you don't have the full series. It's not like you can watch the population every single generation. You're getting it in fits and starts. And by the time you're getting it again, the morphology has changed, you're going to call it a new species. So like it's almost tautological, right? That it necessitates punctuated change.
It's like a friend of mine who took a photograph of her baby every day for the first year of its life. If you looked at the baby every day, you don't see a lot of change. But if you look at the baby at birth and at 1 year, there's a huge difference.
Exactly. Yeah. It looks punctuated. It looks like, wow, that must have happened really quickly. The other key problem is that actually a lot of what Gould and Eldredge wrote about was already predicted in the 1930s and 40s. So G.G. Simpson wrote a really famous book called Tempo and Mode of Evolution, in which he sort of reckoned with this idea that there were stasis followed by rapid change. And he used Sewell Wright's adaptive landscape metaphor. I don't know if you're familiar with this metaphor, but it's basically like, if you can imagine all the possible traits that an organism can have as a landscape, and peaks in that landscape represent places of high fitness, and valleys in that landscape are places of low fitness. And if you just drop a population anywhere on that landscape, then it should evolve towards peaks of fitness, right? And that natural selection is going to push it towards the peak of fitness. And so what happens then is most of the time, populations should sit on peaks of fitness and selection should maintain them on that peak of fitness. Now, when the environment changes, those peaks can shift and they can become in other places. Now, suddenly the population is at a low area of fitness and then it rapidly finds its way to a new peak, right? So most evolutionary change occurs rapidly between these adaptive peaks when the environment changes, while most of the time it's being stabilized on top of a peak. Does that make sense?
Yes.
So that's effectively from population genetics, from like traditional, the modern synthesis, traditional population genetics. That's the explanation for rapid change and stasis. Is that most of the time stabilizing selection maintains you on a peak, and then only occasionally, when the environment changes, does directional selection move between peaks. And directional selection is pretty quick. It's going to rapidly pull you between the peaks when you're thinking in the time scales of geology, right?
Okay. One question I'd like to ask you, which is something we discussed a little bit before the show and is just kind of overarching to all of evolutionary biology and understanding of science in general, is How does one look at a paper and know whether this is a good science paper? Is this something I should read, or is this just a piece of junk?
Yeah. There's a couple of things that and this is, I do this a lot, especially when someone sends me a paper and they're like, what do you think about this? And I can tell you the first couple of things that I do. The very first thing I do is I look at what journal is it in. Do I know this journal? Do I recognize this journal? I don't know every journal, so there's always the chance that I miss something. And if I don't recognize the journal, then I say, okay, does this journal run by some sort of academic society? So there's lots of academic societies, like the Society for the Study of Evolution, SSE, publishes several journals, including Evolution Letters and Evolution. The Genetic Society of America has their own journal. So like, is there some academic society underlying this journal that I can find and read about? And is it like a well-respected society? So those are the two first things I would do. If they happen to pass those barriers, then I also will go to the authors. I look at where the author's from, what is their past work, are they qualified to talk about this topic? And in this case, they wouldn't have passed this, right? Because I think it's a medical doctor and an engineer on this paper.
This is an engineer talking about evolutionary biology. Yep.
Yeah. So this would have failed this particular part, as this person is not known for this. You can also usually Google those people, and I think one of them you can actually find them contributing to creationist work. So you can dig a little bit into them, and then assuming you pass all of those barriers, I then look at who they cite. They're like, are they citing the relevant literature? In this case, they're citing from just this journal. They're citing Dennis Noble. They're citing random philosophers. Like they're not citing evolutionary biologists, right? They're not actually dealing realistically with the material at hand. And that to me is very suspect. The way in which they present the material is very suspect. Talking about things like survival of the fittest, which is a concept from the 1800s. We don't use that like that makes me suspicious. And then when they use phrases like neo-Darwinism, this is another thing that should immediately jump at us as a little suspicious, because neo-Darwinism is an insult term that was used by Conrad Waddington in the 1940s as sort of he used it against a lot of like the modern synthesis, even though it's supposed to be like a gene-centered view of evolution. It's like a critique of reductionism. It's an insult. Then it was used as an insult. So whenever they say neo-Darwinism must mutate to survive, that should jump out at you as this is clickbaity, right? So those are the things. So basically, what's the journal? Who are the authors? Do they cite the relevant literature? And is the journal run by a society, or is it just like some weird pay-to-play thing? You can go to the journal's website, typically go to like submit as if you were going to submit a paper, and read publication costs. Do they have some exorbitant publication costs? That will tell you something about what this journal is really about, right? Like society journals usually have waivers for society members, and society memberships are cheap. So that tells you that this is probably a journal that's going to publish respectable work, as opposed to publishing who can pay the price.
Mm-hmm. Mm-hmm.
So hopefully that's a comprehensive list of ways to evaluate whether this is a journal that you actually want to trust.
Yes, that was very helpful. A couple of things that I should have mentioned earlier. Zach has a podcast and he has a YouTube channel, and there is a link to his channel in the description. So go ahead and subscribe to Zach if you are not already subscribed. And Zach also has a website, which is also linked in the description. So if you have any questions for Zach, if you want to explore some of his materials, have you written any books?
I've not written any books. I do have, I have like my scientific publications that are probably boring and nobody wants to read those. But I mostly, like for my science communication stuff, are all YouTube based.
Okay. So yeah, if you want to check out some of the sources that he has available, those are available on his website as well. So those are some ways to get a hold of Zach. Yeah. We have a comment from Michael Bell who says, he's great and on point. Theists expect others not to check their sources. Almost no one checks clergy, well-known theists. Yeah, that's something I have found to be true as well. One of the things that I have talked about much is I've checked on sources, the J. Warner Wallace sites that they have nothing to do with biology. They're in the area of law. and he gets them all wrong.
Yeah. Creationists are sort of infamous for quote mining, right? It's a thing that they do constantly. They'll quote things out of context. They'll cite, like, one of the citations here in this paper is actually to Stephen Meyer's Darwin's Doubt, which is a book for promoting intelligent design, right? So like a clearly non-scientific book that's being cited in what's supposed to be an academic journal. And again, that's another thing that's super suspect, that those are the kinds of people you're citing immediately, raises a red flag. Actually, Brian Stevens brings up a good point here that predatory journals should be listed as predatory. And there is a couple of avenues for this. I forget the name of the organization there's an organization that basically kind of checks journals, and they write a report every year, every couple of years, on things like impact factors, citation rates, whether or not you have like some weirdness in the peer review process, because sometimes like it's supposed to be peer reviewed, which means they're supposed to send your paper off to experts to review it. But often these predatory journals, like, reviewers refuse to like work with them. And so what ends up happening is the peer review is not real. It's a peer review by the editor that's handling it. And so these investigators like find out, is there any real any realness to it? And then they like, sort of publish a list of journals to kind of beware. But yeah.
Yeah, that's kind of where I started with this article, was looking for peer reviews of this article. And as I said, I didn't find any. When one is looking for peer reviews of a particular article, should you be able to just Google it? Do you need to go to Google Scholar? What's the best way to peer review an article? I should say search.
Do you mean the actual reviewers that read the paper and provided feedback?
Yes. Yes.
Oh, so this is a long-standing issue in academic publishing. Those are normally private.
Okay.
There is one journal, eLife, right now, that has kind of radically transformed the way publishing is working, and they're actually now publishing the reviews with the paper, and that has led to some controversy. But yeah, so there's this whole like weirdness around publishing reviews, because the reviewers are supposed to be anonymous. And there's concern that if we publish the reviews with the paper, then it removes the anonymity, because people will recognize, oh, like, I know that, like, that writing style, I know that, and they can recognize who the person is. And that kind of exposes them or whatever. But there's also an argument, right, to be made that maybe it shouldn't be anonymous, and it should be made clear. So there's a lot of debate in academia right now over peer review and how to properly share those reviews. What I've typically done, what a lot of people have typically done, is just share them themselves. So like, if you like personally want to share the reviews, you can, and you can share them in any sort of public format you want, but the journals typically won't do it, and most researchers don't just share them.
Maya brings up a really good point. Science likes to lock everything behind a paywall, and that's not the fault of the scientists. That's the publishers usually have regulations as to who they will allow to see their publications. And that is why I so appreciate people like Erica Gutsick-Gibbon, who will take those papers that we do not have access to and let us see the full papers.
Yeah, this is, and not to get all anti-capitalist here. There's a real problem in science, in that, like everything in this country, it's about making money. Like at the heart of it, it's about, can I make money? Because the people that control who gets funding and whether papers, like, the journals themselves they want money, right? And so they're selling subscriptions, well, often to universities. Like, most of the subscriptions to these journals are to universities. So if you work at a university, you have access to basically all of these journals, but they come from the exorbitant fees that students are paying so that the university can buy the subscriptions to those journals. So like, there's this, keep that in mind, and that's where your tuition is going if you're in college or you have children in college.
I have wondered that. I have wondered that, because I know that when I was in college 40 years ago, the cost that I paid was a tiny fraction of what I paid for my kids to go to college.
Yeah.
And a big part of it isn't the tuition, it's all the fees. We didn't have all those fees.
Yeah, universities are very bloated in terms of sort of overhead, a lot of overhead bloat, and a lot of those goes to these sorts of things, paying these subscription fees, et cetera. So yeah, it's there's a movement towards sort of open publishing, and this has been led by the preprint world. If you're familiar with the concept of preprints like bioRxiv, all of my papers before I publish them in any journal, I put them on bioRxiv so anyone can freely go and get them. The one thing to bear in mind is that anything you read on bioRxiv has not been peer reviewed yet, but basically you're seeing the form of the paper as it was being submitted. So that can be a useful thing to get to as well.
One last item that was in the paper that I found absolutely revolutionary to me, which I don't know if it's true or not. He was talking about the development of consciousness. And he was saying that the evolution of consciousness is not yet known. I get that. But he was saying that there is some consciousness that is even on the molecular or cellular level. I forget which one he said. That blew my mind that there is consciousness on a cellular level.
So I think the technical word for that is woo. It's the kind of statement that I would expect people like them to peddle. Cells don't have consciousness unless you just want to take the word consciousness and evacuate it of all meaning, right? Molecules don't have consciousness. Prokaryotes don't have that is just like to confuse what the word means, and it's the kind of like Deepak Chopra sort of thing that I would be super suspect of that kind of stuff.
Badger equates it to squids in space.
It is a squids in space sort of thing. Exactly. Yeah.
Okay, very good. Zach, is there anything that I might have missed that you would like to address?
Let's see. I think we actually touched on all the things that I had written down. I guess one last thing is they talk a little bit about what they call complexification.
Yes, I caught that as well.
Yeah, in that they kind of view evolution as this ladder of complexity that supposedly natural selection is driving complexity. This is not the way we understand the evolution of complexity. I talked a little bit about actually how genomes become complex, and it's actually often not through natural selection. It's through mutational processes that are subfunctionalizing duplicates and causing these complicated gene interaction networks to form. And so natural selection tends to favor efficiency and simplicity. So if you want to think about what did natural selection in nature perfect, it's bacteria. Like they are the prime drivers. Like natural selection is proud of bacteria. Natural selection is not proud of us. Like if you think about our genomes, they are big, they're bloated, they are nonsensical, they're redundant. This is not the sort of thing natural selection is very proud of. This is the sort of thing that genetic drift and mutational processes generate.
Not something that an intelligent designer would have designed.
Yeah. Yeah, exactly. There's this saying by, I think it was I don't think it's Brenner. There was a molecular biologist that basically said he would be proud to have admitted to serving on the committee that generated the bacterial genome, that created the bacterial genome, but that he would be very embarrassed and would never admit to being on the committee that generated the human genome. Do you guys have consciousness?
Yeah. Gut bacteria is one of the primary arguments that I use against creationists, because they make the argument that there was no death before the fall. And so I was looking at how fast do gut bacteria reproduce, and how long, if these gut bacteria didn't die, would it take for them to explode our bodies?
Yeah. There's like a law of geometric growth, right? And that if nothing was dying during, the period, I guess, of, before the fall, during the Garden of Eden, then you're neck deep in elephants pretty quickly, right?
I said fruit flies.
Fruit fly, any prokaryotes, are all the way to the moon. Like it's going to be, it would be wild. I've actually seen some creationists try to grapple with this in various different ways. Oh,
really?
It's sort of like a miraculous check on population growth, right? That they just stop reproducing at a certain point by feed it or something.
Probably a lot like the mechanism that stops evolution at a certain point so that you can't cross over kinds.
Yes, this unknown magical mechanism. Yeah.
Well, Zach, I must say it has been an absolute pleasure to have you. It has been illuminating, and I really appreciate your insight and your expertise.
I've had a lot of fun. Thanks for having me on. Yeah, this was enjoyable.
And if anyone is not already subscribed to Zach, please subscribe to Zach. And next week, the guest will be John Hammond. So make sure you come back for that as well. And to all of you in the chat who contributed comments and questions, thank you very much for your contribution. To all of you who are viewing the future, thank you for the view. And especially thank you, Zach, for coming.
Thanks for having me.
Live your life.